I'm very pleased to introduce the next speaker, Professor Caldas from Cambridge as well. He holds the chair of cancer medicine at the University of Cambridge and heads the breast cancer functional genomics laboratory there. And his main research focus is on functional genomics of breast cancer and its biological and clinical implications. And now we are mainly back again to somatic mutations, sporadic tumors, and you are speaking about stratification of breast cancer into 10 genomic driver-based subtypes and its clinical implications. Please, Carlos.
Yeah, thank you. So first, thanking the organizers, in particular Christoph, for inviting me. It's always a pleasure to be back here at DKFZ. Second, apologizing for not being a germline cancer geneticist. But I think, what I would like to preamble my talk to is to say two things. One is that as important to know what your risk of breast cancer is, you want to know what is the risk that you'll get a bad breast cancer, okay? Because as I'll show you, there are many types of breast cancer and some of them with very long-term survival. And then the second is, are there more of these genes to be discovered? And what I would like to say is that I am certain that there are, although we haven't found them yet. And if you stratify, I'm sure you'll discover them. And as you saw from Doug, with these very large studies, you're really only accounting for ER positive and ER negative. As you could see, most of the discoveries are in ER positive. So you are not even including HER2 testing, which is something that we do routinely in the clinic. So immediately you're saying with this that there are only 2 types of breast cancer, and so you will have your discoveries limited in that way. We know now, by looking at many cancers and looking at their genomic landscapes, that really there are 2 types of somatic events that dominate these genomic landscapes. One is copy number aberrations. And we heard a lot about sequencing today, and people seem to forget that there are other types of aberrations in cancer genomes that you would not necessarily pick better by doing sequencing. And that is amplifications of oncogenes and hemizygous deletions of tumor suppressor genes, a lot of which will not be accompanied by mutation of the second allele. So you would not discover them by sequencing. You might discover them by epigenomic sequencing or profiling. The second event is, of course, point mutations, which is what most of us are now concentrated on because of the advent of next-generation sequencing, which is getting logarithmically cheaper. It's even surpassing Moore's law, if you want, as our ability to do it at a very cheap price. And we're soon going to have the ability to do exome sequencing for about 100 euros per sample, okay? Not whole genome, but whole exome. So in this effort, Chris Sanders' lab, and at the time there were data for about 8,000 tumors in TCGA. Now we have data for close to 20,000, and the data would not change. The genomic landscapes are dominated by these 2 events. And they looked also at microRNAs. They contribute very little. And they looked at methylation in a limited way, using Infinio arrays for Illumina that mostly look at promoters. And methylation events profiled that way contribute very little. But what is striking is this observation, is that if you look now at the mutations, if you want, as the somatic aberrations, and you divide them into the 2 classes, copy number aberrations and mutations, you see very nicely that breast cancer is only surpassed to it by ovarian cancer in having its genome dominated by copy number aberrations, not by point mutations. And actually, this is real data. You rarely see this in biomedicine. So this is data from 7,500 cancers thereabouts, but with 15,000 it doesn't change. The little squares are shades of blue. The darker, the larger the number of samples. So you see that for a lot of these squares you have data from a lot of tumors. And if you look at recurrent copy number aberrations, where you would expect oncogenes and tumor suppressor genes to be, and recurrent mutations, where again, in the point mutation space you would enrich for oncogenes and tumor suppressor genes, you see that there are no tumors in this quadrant. In other words, there are no human tumors that have lots of recurrent point mutations and lots of recurrent copy number aberrations. There is actually a very nice anti-correlation. And if you look genome-wide, even if you don't stratify breast cancer, so this is from our paper in Nature in 2012, looking in this case at 1,000 cases, when we look at the second 1,000, we saw exactly the same. You don't stratify the tumors, and you just look in gene expression space for outlying expression. And that is very simple. If you look across tumors, most gene expression will have a near normal distribution, it will have a Gaussian distribution. So you then look for non-Gaussianity, and you look for outliers. And then you see the outliers that co-register in cis with copy number aberrations. And if you do that, you can resolve the apparent bewildering complexity of copy number gains and losses into about 45 or so loci. And in these 45 loci, you have in red oncogenes and in blue tumor suppressor genes. And even if we didn't know this, but we knew, of course, it's nice to have as a positive controller B2, we would have discovered it. There are several new oncogenes here, some that we know about, some that are new, and several tumor suppressor genes. And again, we might not have had any ability to do sequencing, we would have rediscovered P10, for example. It was screaming in the data on chromosome 10 that there were deletions there and that there were some homozygous deletions associated with no expression of P10. So it's quite obvious that if you combine copy number and expression data based on this prior biological knowledge, if you look at genes whose expression is driven in cis by copy number aberrations, by definition, you will be enriching for oncogenes and tumor suppressor genes at those loci. And so that's what we did. We picked the top 1,000 genes. Again, we could have picked 200 or 2,000. It didn't change that much, the final result. This gave us the more robust and reproducible result. Of course, we don't know what the ground truth is, but the solution that was most parsimonious and therefore the closest approximation to the ground truth is that doing this, the tumors fell very nicely into 10 clusters or 10 classes and hence 10 diseases. And if you see, this is not a classical heat map of either gene expression or copy number aberration. This is a heat map representation at each of the 1,000 loci in 1,000 tumors of the scaled product of gene expression and copy number values, which is a way of summarizing this information that the expression is driven in cis by copy number aberrations. And so, for example, I'm not going to have time to go into all the classes. You see here these tumors, which are ER-positive tumors, HER2-negative. This is about 5% of breast cancers. These tumors have horrible prognosis. They are defined by an amplicon on chromosome 11q13. They're completely different from these tumors, which are also ER-positive, HER2-negative. They are driven by an amplicon at 8p12. So these are two different molecular diseases. And indeed, you can say, well, you had 1,000, you did this very carefully, you look at co-clustering, clustering, reproducibility, shadowing, all of the methods that we could discuss if you wanted ad nauseum. But how do you know that this is really an approximation of the ground truth? So we had a further 1,000 tumors. We applied exactly the same classifier. And what it shows here is the first 1,000 tumors, the second 1,000 tumors, divided into the 10 classes or integrative clusters. And indicated on this panel in red and blue are frequency plots of the copy number gains in red, the copy number losses in blue. And if you look across the 10 subtypes in the first 1,000 tumors and in the second 1,000 tumors, they are identical. In other words, we have discovered 10 classes that are reproducibly present in the data. This is very robust statistically using this statistic proposed by Bob Tibshirani. And they have very similar compositions of intrinsic subtypes, but they clearly surpass the intrinsic subtypes. And for example, just out of one example, you see here in red basal-like breast cancers. Yes, a fraction of them, about 3/4 or so, are very genomically unstable. 90% of these tumors will have a p53 mutation, but there are other basal-like tumors in other classes, and in particular, if I look here in tumors in this class, which are genomically very quiet in cluster 4, about 1/4 of these tumors are also triple negative. About 60 to 70% have p53 mutations, so not as much as in cluster 10, but they are a completely different genomic entity. And by the way, these tumors usually have very heavy lymphocytic infiltration and suggestion that women have elicited an immune response. We have now validated this quite robustly So this is 7,500 samples that are external from 40 different studies for which only gene expression data was available, with the exception of the TCGA data, which is here. And we produced a classifier that you can use. It's available on R and Bioconductor to classify the tumors into the 10 classes based on gene expression alone. And you can see that in these 7,500 tumors, you find exactly the same 10 classes, and they have pretty much superimposable outcomes to what we reported in the Nature paper. Now, this is unpublished data to show that these remain entities that are distinct, and if women are unlucky enough to develop metastatic disease, they remain so from the moment of diagnosis until the moment of death. So this is now, because we had the complete outcome of these 2,000 women, and this is a cumulative Kaplan-Meier to show that, of course, the worst clusters have more metastases and the better clusters have many fewer metastases. But even after you diagnosed with metastatic disease, you see that there is marked separation between these subtypes. And so, for example, tumors with the better prognosis, ER-positive ones, which is one of the clusters, these women with metastatic disease, half of them are alive 12 to 15 years later, okay? So it's important to classify into these classes. And it's important because even if you look at a gene like P53, like PIK3CA, the most frequently mutated oncogene in breast cancer, and you look at the effect of mutation in this gene on survival, you see that it is dramatically different depending on your copy number subtype. So here we're looking just at ER-positive tumors, quite a lot of them, with the exception of a small number that are in cluster 10. So they behave like triple-negative breast cancer, by the way. About 75% of these tumors are ER-positive, but they have a P53 mutation. They are very genomically unstable. You see that in the good prognosis clusters, which are 3, 4, 7, and 8, if you have a PIK3CA mutation in these 1, 2, 3, 4 clusters, it doesn't appear to change your prognosis. If in anything, a PIK3CA mutation confers you a better prognosis than if you are wild type. Whereas if you look at also ER-positive clusters, 1, 2, 6, and 9, which are bad prognosis ER-positive clusters, if you have a PIK3CA mutation, you do even worse. So in other words, just with one gene, depending on what subtype you are, so what disease you have, PIK3CA will have very different prognostic impact. And so, based on this, we used the papers published in Nature. We couldn't afford to do exome sequencing at the time in 2,000 cases, so we picked 178 genes. It's about 1 megabase. These genes are, of course, genes that are known already to be mutated in breast cancer, because a mutation had to occur in at least 2 samples from these studies published in Nature for us to select the gene. And so we've sequenced these genes in 2,500 cases, classified into the 10 classes, and we sequenced them at depth, you know, somewhere between 100 and 500-fold. And so what you see is what I told you, that even in these genes, despite being enriched for genes that are breast cancer genes, you see that you don't have many mutations. This is very different from colorectal cancer, melanoma, lung cancer, renal cancer, etc. As I said, the landscapes are dominated by copy number aberrations. With a few exceptions, you know, very quickly you fall into a tail. There are only 3 or 4 genes that are mutated in more than 10% of the cases. There are only, you know, 20 or so that are mutated in more than 5% of the cases. So using purely the data, because we had 2,500 cases, people can get into statistical discussions. We just followed a very pragmatic approach proposed by Bert Vogelstein in 2013, which is a ratiometric method to look at genes, and based on the pattern of mutations, not on the frequency, but on the pattern, you classify them and you give them an oncogene or a tumor suppressor gene score. And when we did that, we identified 40 mute driver genes, as Vogelstein called them. And here they are. I don't have time to go into the details. But if you look now, there are about 2,200 tumors here for which we have copy number and mutation data. You see the number of peaks that they have on their copy number space. the number of mutations that each of the tumors has in the 40 driver genes. And as you see, there are no dramatic differences across the 10 subtypes, but what you see is a very different pattern of these mutations co-occurring or being mutually exclusive. And you see clusters with lots of mutations in AKT pathway genes, in particular PIK3CA, and very few mutations in P53. And you see all kinds of combinations if you look at the data. This has impact also where you find the types of mutations that Doug was talking about. So let's start, for example, with BRCA1. So these are germline mutations now. Look at the scale. So you are talking about half to 1 or 2% of the tumors. But when you find mutations in BRCA1, you see them mostly in the tumors that I told you are 90% P53 mutant, triple negative, very genomically unstable, but you also find them in triple negative tumors that are genomically quiet and have fewer P53 mutations, and then salt and peppered across clusters with the exception of the very good prognosis ones. There are only a couple of exceptions there. For BRCA2, it's much more dispersed story, if you want. Same for CHUK2. For P53, again, very rare, even rarer than BRCA1 and BRCA2, but they mostly in aggressive tumors, either triple negative or HER2 positive. Because we did deep sequencing, we could also look at clonality of mutations, and the majority are clonal, so they exist in 100% of the cancer cell fraction. And we determined this using the method proposed by Charlie Swanton's group, published in Science Translational Medicine 3 or 4 months ago. But you see here clear examples of subclonal mutations in drivers. These are all primary breast cancer. So here is, for example, a GATA3 subclonal mutation or an ECADERIN subclonal mutation there. And really, you can look at this and look at whether there are more or less clonal mutations depending on whether the genes belong to the 40 drivers or to the remainder 140. And we could spend time on this. There is nothing that jumps at you on the data, but there is a tendency for clonal mutations to be more common in drivers. And what is very interesting is that there is a quasi-linear correlation between chromosomal instability measured as a fraction of the genome that has copy number aberrations and intratumor heterogeneity in mutation space. And you see here that there are grossly 3 classes of tumors, but it's not always what you would expect, because these tumors here, which are the very bad prognosis ER-positive, HER2-negative tumors that I told you in cluster 2, they have few copy number aberrations other than the defining amplicon at chromosome 11q13 and very low intratumor heterogeneity. Now, I don't have time to go into immune response, but suffice it to say that it varies dramatically across these subtypes and that we see evidence of immunoediting, in particular in the triple negative tumors with quiet genomes that I mentioned to you. There you see evidence of immunoediting because you see clearly that there are less observed than expected neoantigens when you sequence these genomes. There are different drivers here, but what I hope to have convinced you is that we really need to start thinking of breast cancer as 10 diseases with distinct copy number aberration drivers and different profiles of mutations. These are the relevant papers. These are the people that have done the work, a lot of it in collaboration with Sam Aparicio at the British Columbia Cancer Agency. And I talk, and they seem to be smiling, and I do most of the work. Thank you.
Thank you so much, Carlos. Yes, the talk is open for discussion. Maybe a first one from me. What would you think is more predictive on the subtype if you go for the BRCA1 and 2 tumors? You showed, I mean, a BRCA1 tumor and a BRCA2 tumor, both showing the same cluster. Are they more similar than the BRCA1 tumors with respect to disease cause and response to therapy and so on?
Yeah. So That's a very good question. So, by the way, these are just germline mutations. We identified unequivocal somatic mutations in BRCA1 and BRCA2, very small numbers, but we clearly identified them. You know, even in a very large data set here, like this, you're talking about very small numbers. And so, to answer your question, you need to have bigger numbers. But as you know, in germline BRCA1 and BRCA2 carriers, in ovarian cancer, there is a very clear synthetic lethal story with PARP inhibitors. But as you know, that's been more difficult to prove in breast cancer. And I don't have data to show you today, but we have data from xenografts that we have from a significant number of triple-negative breast cancers, including from BRCA1 germline carriers, that the synthetic lethality story in breast cancer is not as clear-cut as it is in ovarian cancer. So just give you one example. So I think it's complex, and I think you're going to need more numbers. But the point with these very highly penetrant, high-risk genes and the distribution from the subtypes, the point that I make is, and to go back to what my preamble, is that I think we're going to need to do GWASs in some of these subtypes and we will identify more predisposition genes. And I think, of course, as I said, that what we want to know is you predisposed to what type of breast cancer, because some of these are like chronic disease. You know, you have survival at 10 years in cluster 3 tumors of over 90% survival at 5 years and over 85% at 10 years in a lot of these patients treated with modern surgery and hormone therapy. Yeah.
Oh, this adds another complexity to the hereditary causes. Further questions? So, yes, over there. Everybody is overwhelmed from your huge data sets. Yes, please.
How do the integrative clusters, have you looked at how they correspond to the different types of differentiated and stem cells in breast tissue? How do they correspond?
Well, I don't like the term cancer stem cells. I like more the term tumor-initiating cells. But you can say it's just semantics. But each of these tumor subtypes will have a particular population of tumor-initiating cells that will be different from the other subtypes. That's my hypothesis. We'll now have the models to prove this. We have now about 80 xenograft models, and we have representation of all clusters with the exception of one, which are cluster 3, the more indolent ER-positive tumors. Some of these xenografts take about a year to grow in the mouse. The very indolent ER-positive ones. So eventually, with cell lineage tracing studies with lentivirus, etc., we'll be able to answer that type of question. But we have the tools now to do that. Yeah.
And one last question here, Dr. Brenner.
Very interesting data question. Are there empirical data showing differences between screening detected cancers and other cancers, yes, with respect to the distribution of these 10 types?
Yeah. So we don't have enough data even in these 2000 to be able to tell that. We have, with Paul Farrow and Gordon Wishart when he was one of our surgeons, and Sarah Jane Dawson, one of my clinical fellows, we looked in search and we did with immunohistochemistry some stratification of tumors and looked at screen detected versus interval cancers, and there are differences there clearly. Most of the cancers picked in the screening program, my bet is that they're going to be in clusters 3, 7, and 8, which are the better prognosis tumors. And whether we will be able to pick, for example, tumors in cluster 2, which have horrible prognosis, and whether they will have different markers is a question mark, as it is for clusters 1 and 6 and 9, which are equally aggressive ER-positive tumors. But we don't know the answer. So we're trying to produce a test that will be very cheap, available on the National Health Service, that we could do in a routine pathology laboratory. We're doing that as we speak, and maybe we'll have results within the next year or so, and then that there will be a test that could be done cheaply, and you could classify tumors based on routine pathologically collected material, that is paraffin-embedded blocks.
Maybe one last very brief question. Do you see shifts between the clusters during prognosis and resistance development?
Okay, that's an excellent question. So the answer is, but based on a small n, so it's got a caveat, the answer is that we see that very uncommonly. Now, we have more data for neoadjuvant therapy, where we have serial biopsies, and with the exception of women that have pathological complete response, so there is no tumor there by definition, we don't see migration between these. So the way I like to look at this, and I skipped over that slide, is that if you want, you can look at this cluster. So all tumors have intratumor heterogeneity, of course. And so you can look at them as trees, if you want, to correspond to phylogenetic trees. And some of these will be eucalyptus, some of them will be olive trees, some of them will be oak trees, etc. And so they are families of trees that will remain like that. Now, of course, a eucalyptus, if it's closer to water and it's got more sun, might grow taller than another one, but the shape of the phylogenetic trees will be similar. Now, we need more data for me to be able to substantiate what I just said, but all of the preliminary evidence that we have points in that direction. So this is not hopeless. It's not like one olive tree can become a eucalyptus a year later. No, an olive tree will remain an olive tree, if I can use that image.
Yeah, thank you very much. This was a good last sentence. So thanks a lot again, Carlos.